Hello everyone. I made this PowerPoint video to describe how we do gene calling, which means we're going to assign the appropriate start sites for each of the genes in our chosen genome. So first, let's start with the central dogma of molecular biology, which says that the flow of genetic information is from DNA to RNA and then RNA to protein, and that occurs in all cells. So if we have the sequence of the DNA, a strand of the DNA is used as a template to transcribe messenger RNA, and then the messenger RNA is used as a template to translate into protein. So we have protein coding genes in our genome, and we want to find out what those proteins are, and we want to find out where the start sites are for all of these genes. So we're going to be given in DNA Master, we're going to be given a sequence of DNA, and I just wanted to remind you the DNA is a double helix, and that's shown here in blue, and the two strands are anti-parallel, so we go 5' to 3' and here, and 5' to 3' in this direction here. And during transcription, the strands of the DNA separate, and we use one of the strands as a template to make messenger RNA. And this down here, we're using the bottom strand as our template strand, so that's the non-coding strand. So you can see that because the messenger RNA is complementary to the bottom strand, and the top strand is also complementary to the bottom strand, that the sequences and the directionality in terms of 5' to 3' are exactly the same. The only difference here in the sequence is that we replace the T's with U's. So here we have ACA, GC, T, TA, and here we have ACA, GC, GU. So when we import our phage DNA sequence in DNA Master, we'll only be given one strand. We're going to be given the top strand. We're going to use the strand that we don't typically, or for most of the genes, we don't use as a template. And we'll be reading it in the 5' to 3' direction. So when we say gene number 1, it will be the one on the 5' end. So any genes that are transcribed and where we use the bottom strand as our template and we make messenger RNA, we're going to say that we're transcribing those genes in the forward direction. There are genes on our phage that will actually use this top strand, but because the messenger RNA has to be complementary and anti-parallel, that means that they will be transcribed in the reverse direction. So we'll refer to this as forward in reverse. And in our genome, the vast majority of our genes are transcribed in the forward direction. So we want to create a good set of notes. So I'm going to go through all of these so we can see what they are. So the first one is SSC, which is start-stop coordinates. So there's this template that we imported into DNA Master, and we are going to call the start at the 5' end as 55 and the 3' end as 444. So we're going to say start 55, stop 444, forward. So we're going in the 5' to 3' direction. The next thing we want to do is we want to assess coding potential. So I'm going to get out of my PowerPoint now and go to my coding potential maps. So also, I'm going to be illustrating how many tabs you have to have open when you're doing this. So remember we ran GeneMark on this, and this would be our self map. This is our SMEG map, and this is our tuberculosis map. So if you go in the SMEG map is nice because it has all the GeneMark calls on it. So you'll see all 6, you can see here I'll make this smaller so you can see, and then I'll make it bigger again. You can see that we have all 6 reading frames. So we have the top 3 are the top forward, and the bottom 3 are the reverse genes, and there'll be a black bar where GeneMark has called the start of the gene. So coding potential just means that that the program has looked at the sequences of the bases in the DNA and think that it's likely that there are genes there. So if we look at this, we can see that the The line is the open reading frame. That's a stop codon, and all of these little things going up are all possible start codons. So remember, we're going to call the start at 55. GeneMark called the start at 151, so that's why this black bar doesn't go all the way to 55, because that was a glimmer call. But you notice that this is why we're not calling the start at 151, because we miss coding potential. So we don't want to do that. So that's our self map here. If we look at our host, because the coding potential is going to be similar. And again, here's that same gene, and there's our 55 start. Again, if we started a little later, we'd miss coding potential. And for tuberculosis, same thing. So our start at 55 is going to cover all the coding potential. All right, now I'm going to go back to my PowerPoint. And so this is what I'm going to say in my notes. So CP is coding potential. So I write CP : start covers all coding potential as shown on self, SMEG, and TB GeneMark coding potential maps, and then I write a semicolon. Okay, so we're done with that. Okay, what's the next one? The next one is SCS. I think that's Start Choice Support. I was actually not able to find that on C-Pages, but I'm pretty sure that's right. So there would have been a note when we did our auto annotation, so you want to cut and paste that. So for gene 1, it says original glimmer call at base pair 55 has strength 11.93. GeneMark calls the start at 151. So the way that this works is that GeneMark here did disagree with Glimmer. If GeneMark agrees with Glimmer, GeneMark won't be mentioned. For example, if GeneMark happened to agree, you would just see original Glimmer call at base pair 55 has strength 11.93, and then that would be the end of that that remark. So GeneMark won't be mentioned if Glimmer agrees with GeneMark. If GeneMark disagrees with Glimmer, it will be noted, and that's because we set up our preferences to look at both calls, but to favor Glimmer calls. Okay, Starterator is next. Okay, so if you, I have a link here that shows a video of how to interpret Starterator data. So that's a really great link. So here, so I'm hovering over this so that you can see what the link is. But you can access this PowerPoint so you can see the link here. So what I'm going to do is I'm going to go to phagesdb. So again, I'm going to get out of my PowerPoint now, and I'm going to go back to my browser, and I can see I've already done LandGRX, so it comes right up, and then I click this green arrow right here, and I have a lot of information here about LandGRX, but one of the things I want to do now, this is LandGRX phage page, is I want to go to the gene list. So I'm just going to click here and go on gene 1. And when I do that, I have information about gene 1. So there's the FAM. So remember, we take all of these genes and we put them in families. So it's FAM for short, and it's PH because of phage. But this gives us access to the Starterator report. The other thing we might want to look at is if we can look at other members. There are 172 members of the same FAM, and just like as a little hint, it also gives us some information about function as well, but we'll talk about that later. So what I want to do now is I want to click on the Starterator report, so I can just view this in my browser. So I'm going to open this up. So you would have seen in the video the explanation about how these all of the members of this FAM are grouped into tracks, and how all of the possible start sites are given a number. So what I'm going to do here is I'm just open my Finder, and I'm going to put in LandGRX so I can see where LandGRX is. This is a 26-page document, so sometimes it can be difficult. So this is on the second page. So this shows that LandGRX is in track 54, and there are 8 other members of LandGRX. And I'm a little confused as to why this isn't yellow. In the video, you heard that if you see a start that's green, that means that it has actually been annotated, that somebody has gone in and looked and verified that that's the correct start site. So it seems like since we're a draft, it should be yellow. I'm not sure why it's not. But this is, out of all the possible starts, this is the 10th possible start. So what Starterator does is it goes all the way back to the beginning. So this is base pair 55. So it goes all the way back to base pair number 1. If this were not the first gene, if it was the second gene, it would go back to the sequence in between genes, and that's the 10th possible start site. So if we continue our finder, we can see here's track 54, and these are other phages that also are in that track. And these are also the genes that call this the most annotated start. So we can see that Landrack that the start is called there, but that's the most annotated start. And if I continue with my finder, again, this is start 10. Again, we saw that there were 172 members of that fam. So this is found in 43 of them. So that's not a huge percentage, but it is called 95.3% of the time when present. So again, these are all the same phages that have the same start as Landrack. And down here, we can see start number 10 starts at base pair 55. And this tells us that this start has 37 of the members call this the most annotated start. So again, that's not really high, but the only other possibility, and that was the gene mark call at 151, that's start number 39. So again, if you're numbering all the starts, has only one most annotated. So it's pretty clear here that start iterator calls the start number 10 at 55. These are other possible starts, and it will give us the start number plus the base pair number, but none of these have been called. Okay, so if I, so I have all of the mentions of Landrack here. So I'm going to go back to my PowerPoint. And so what we do for this part of our notes is that we say that, are we going to use the suggested start? Maybe we disagree with start iterator and we'll say that's not the suggested start, or maybe it's not informative. Maybe if we look at start iterator, we see a wide variety of different start possibilities, but start iterator doesn't really agree on them. So so here start iterator pretty clearly does favor the 55 base pair call. So we'll write suggested start. So again, if I go back to my notes right here, where am I? Okay, so ST is start iterator, and I wrote SS for suggested start. Okay, so BLAST data can support your start. So if there's good alignment with other similar genes and similar phages. And again, we have 172 members, so we should be finding some of these hits. So you should, in your notes, list the phage name, the gene number, it's not always provided, the QS coverage, the E value, the percent alignment, and percent identities. So I'm going to go out of my PowerPoint and into DNA Master. So let me minimize all of this and go into DNA Master. So I'm on gene 1 right here. Let's see, I recently made some changes. I'm going to go ahead and hit post to make sure that my changes are there. So if I'm on gene 1 and I go to BLAST, let me make this window a little wider so we can see what's going on. So these are our BLAST hits, and there are 2 tabs down here. So I like to look for alignment. So what we have here is the sequence of the protein for gene 1 of Landrack is at the top, and that's called the query. So that's Q for query. And then here we can see this is phage Bartholomew. They don't actually tell us what gene number it is, so we won't be able to do that. But we can see that the sequence of this gene in Landrack is identical to the one in Landrack. So they have, and it's lined up where the first amino acid of Landrack is the same as the first amino acid in Bartholomew. And if I look at Jebex and Fishburne, I see the same thing. So what I like to do is you have a lot of hits here. I usually like to report the top 3, unless there's another reason not to, but I like to report the top 3. The other thing is if you click on this tab, it will give you some information in terms of the percent identity, percent aligned, and the E value. So percent identity means 100% of the amino acids are all the same. So this is a 129 amino acid small protein, and all of them are the same. and they're aligned exactly, again, where the query is number 1 and the target is number 1. And from 1 to 129, they're completely aligned. So that's true for Bartholomew and for Jebex. If I go to Fishburne, I can see that there's 100% alignment, but 123 of the amino acids out of 129 are the same. So the percent similarity is 95.35%. I think that is different than what I saw before. So let me look at that. Okay, and if we see that, so we can see that wherever there's a line, that means that the amino acids are the same. If there's a plus, that means it's similar. And I just noticed there's some X's right here. So I think what that means is that in order to get the rest of these to align, that amino acids had to be skipped in order to do the alignment. So the alignment here isn't perfect. Or to do the alignment, you have to do it in a way that the similarity is not 100%. But the Q1S1 tells me that I've called the start correctly. So okay, so 95.3%. So I'm going to go back to my notes. I'm not sure why I typed that before, but I'm going to do 93.35%, is that what it was? Okay, hit post. Let me go back and make sure I got that right. All right, why did that? Okay, it's right there. Oh, that's 95.35%, similar, 94.5% identical. Yeah, let's do the identical, not the similar. Okay, so that was 94.5% identical. Okay, so 94.5%. So the way I write my note then is that I say blast start Q1S1. So you write Q1 colon S1 for Bartholomew, Jebex, and Fishburne. They're 100% aligned for all. The E value is 0. So the E value tells you how closely related they are, and in terms of, is this an actual similarity or is it due to chance? And the closer the value to 0, the more sure you are of this, and the less likelihood this is due to chance. So an E value of 0 is actually perfect. You'll see lots of really, really small numbers that are close to 0, but this says the E value is 0. Okay, so E value is 0, 94.5% identical or greater. So I do this because the Jebex was 94.5% identical and the other ones were 100% identical. So again, this comment is going to help me support my choice of 55 base pairs. So let me here put post to make sure my changes are taken. All right, so let me go back to okay, so for gene 1, we have Q1S1, 100% aligned, 94.5% identical or greater, and an E value of 0. Okay, what information do we also need? And that's the gap. So that's the number of bases between the beginning of this gene and the end of the last gene. So the beginning of this gene is the 5' end, and the end is the 3' end. So this is not applicable for gene 1 because there is no previous gene, but if we would go to gene 2, we would list the gap as 32 base pairs. And we would use it as a negative value because we're going to use the positive value if there's a gap and negative if there's an overlap. So let's go back to DNA master, and I can show you this for gene 2. So here we see we start at 55 and end at 444. For gene 2, we start at 413. So if this ends at 444 and this starts at 413, then we have an overlap. And if you do the math, I'll go back to my PowerPoint. If you do the math, if you took 413 minus 444, you'd get minus 31. But the way these things work, because it's a gap, and the way that you count, then you would add a base pair for an overlap, and you would subtract for a gap. Okay, longest ORF. How do we do this? So longest ORF, is this the longest open reading frame? Okay, let's go back to DNA master. So we're going to use something called DNA frames. So if I go to DNA and then frames, then I get all 6 of my reading frames, and then I hit ORFs. And I actually like to blow this up a little bit so I can see what's going on. And there's no scroll bar, but you can click on these arrows to go back to where you want. Maybe I'll make this a little bit bigger. Okay. So there's our gene number 1 that's in this top reading frame, and gene number 2 is actually going to go down to the second reading frame. So the phage will use these various reading frames. So what I want to do is, if I look at gene 1, the lines going all the way up to the top, those are stop codons. And all of these going halfway up are all start codons. So again, that's our job, is to figure out which one of these is right. So I'm going to take my mouse and I'm going to click right here. And when I do that, I get this nice line. If sometimes it's trial and error, so if you don't get it right, just hit the undo button and try again. Like if I did this, for example, like if I did that wouldn't be right, because I want to highlight the whole reading frame and I want to get all the start codons. So I'm going to hit the undo and try again. So if I click over there, I get the whole reading frame. And now I'm going to click on ribosome binding site, and I get another window, and it tells me all the possible starts and the length of the ORF. And you can see that if we are going to call our start at 55, that is in fact the longest possible ORF. So if I go back to my PowerPoint, so you just say yes or no. Is it the longest ORF or not? And we would say yes, this is the longest ORF. And here is the a screenshot of the window that I get that shows that. So you can watch this translation video on your own, but just as a reminder, we're using messenger RNA as a template for protein synthesis. So we call that translation because we're translating the bases into amino acids. And when we have our messenger RNA in the ribosome, we have to align the messenger RNA properly so we get the proper reading frame. So if we have the messenger RNA aligned, we're going to have 3 possible reading frames, and we want to get the right one. So this is a whole bunch of genes just sort of lined up so we can see the similarity. But if that's our start codon right here, and if that sets the reading frame, the way we do this is that there's a sequence upstream that is complementary to a sequence on the 3' end of the 16S ribosomal RNA. So we need to, if you remember from your general biology, the messenger RNA locks into the small subunit of the ribosome before you add the large subunit. So this is called the ribosome binding site, or maybe Shine-Dalgarno sequence after the people who discovered it. So it's shaded orange in here. So again, that sequence is complementary to a sequence shown in green on the ribosomal RNA. So the purpose of this is to set the proper open reading frame and to find the initiation codon. So I have another picture right here that then again shows a whole number of genes right here. And this actually shows how we have, this is the small subunit on the ribosome. And if we have this nice base pairing right here, then we can just kind of look and say, oh, there's the first AUG. That's going to set my reading frame and my, my initiator tRNA combined. So this is called the Shine-Dalgarno sequence, or the ribosome binding sequence. So what this program does is it looks for every AUG in every reading frame and looks ahead to see if there's something that looks like a Shine-Dalgarno sequence. If I go back to that box that was pulled up, I have some data right here. You should make sure that you're set on Kibler 6 and Carlin medium, just so that we use the uniform system with everybody else in the C-phages program. I do have a link for how to interpret the data, so I'll go ahead and mouse over the link so you can see it. What we have is a raw score, a z-value, a spacer distance, and a final score. And I'm going to look at the values for our chosen start, which is 55 base pairs. So these values logarithmic. The raw score and the final score are both logarithmic, and scores that are closer to 0 are better scores. So our raw score is minus 5.021. Our spacer distance is 9. So that just means that, if I go back up here, we're counting the number of base pairs from the Shine-Dalgarno sequence to the start of the gene. So for this particular Shine-Dalgarno sequence for gene number 1, the spacer distance is 9. And you can see there's a variety of spacer distances. The final score integrates both the Shine-Dalgarno sequence raw score and the spacer distance. We have a final score, and the genomic Z value, that's the standard deviation. So this isn't the best score because you can see that there are other values that are closer to 0 than this one. So, and there's our ATG start codon. You know, for phages, sometimes we use GTG instead of ATG, or maybe even TTG. So the ribosome binding site gives us some information, but we shouldn't choose the start based on Shine-Dalgarno sequences alone. It's just something for us to note. So what we'll do on our notes is we'll report the raw score, the spacer, the final score, the Z score, and then we'll just note that it's not the best score. Okay, finally, function. We want to assign a function if we can, and there are functions present in all genomes that we can look for. So I'm actually going to follow this link right here. So if we this will give us take us to the C phages bioinformatics guide here. So So what we need to do is we need to look, because there are functions that must be present, that all phages have them. There are some that are found in most, but all genomes, and there's some that might actually not even be identifiable. So we have, so you can go back and read this, but it has a list of all of the functions, and then this is the rank in according to whether do we find them in all of our phages, or some of them may or may not be identifiable. So there's that. The other thing is, and this is really, really important, this is the approved function list. So I'm going to click here. So that's also from the C phages guide. And we should also note the last time that the functional assignments were updated. So here it's actually kind of recently, so on February 4th, 2022, that's when this was updated. So we will be, if we look at this left-hand column, we will be told these are the functions that we can use. These are some old functions names that we should not use, and there will be some notes that will help us out in our assignment. So what we want to do is we want to use a number of lines of evidence to assign function, if we can. Sometimes we can't assign a function. So we're going to use BLAST, HHPRED, Famirator, and the concept of Syntony. Okay, so the next thing in our notes is SIF BLAST. So SIF is supporting information for function using BLAST data. So here, if I go back to my DNA master, and I'm going to have to close out these windows since I'm not using them anymore. So again, here I'm in gene number 1. If I go to BLAST, you can see that for these genes, these phages, sorry, Bartholomew, Jebex, and Fishburne, that terminase small subunit is assigned for all 3 of them. So in my description over here, so I have function SIF BLAST, terminase small subunit for extra space here. Let me get rid of that. For Bartholomew, Jebex, and Fishburne. Okay, so I'm going to be somewhat confident, because I need to look at all of my lines of evidence, that this is a terminase small subunit. The next thing I need to do is I need to look at something called HHPRED. And what I'm going to do there is I'm going to go over to the tab where it says product. So that's the protein that this gene makes. And what I'm going to do is I'm going to highlight this. Now the commands in either your Windows machine or your Mac don't work here. So what I'm doing right here is I'm not using Control-C or Command or whatever. I'm right-clicking, and then I'm going to choose Copy. And then the next thing I'm going to do is I'm going to go to a site called HHPred. Now I already ran this, but I'm going to show you what to do. Is you go to, I'm just going to Google HHPred, and this should come up. And the other thing I can do is I can sign in, and it remembers my password, thankfully, because I do not. So I would encourage you to sign up and get an account. It's free. And then I'm going to do the same thing. I'm just going to right-click and then hit paste. And then if I'm doing a lot of these, what I like to do is I like to give it a name. So I'm going to call this. This is Langerak, because I do a lot of phages. This is GP1. Needs to, I think that's enough characters. And then I'm going to hit submit. So it says your submission is being processed, and that could take a while. So that's why I like to name these. You can see I've been doing some other things over here. And if you're logged in, if you want to go back to something, this is another phage I was working on with another student. I don't have to run it again. I can just go back to it. But I did already run this ahead of starting this video, so we wouldn't have to wait. And here are my results. And what I see here is the way that the colors work is red is good, and as you go through the rainbow colors, then it's less good. So we can see that in terms of the alignment, this is 129 amino acid protein. So we're aligning pretty well from say 6 amino acids, 16 to 115. And here are our probabilities. So one of the things we should note for the C-phages program is here we have probabilities greater than 90%, and then it goes to 50%. So you should never write down anything for HHPred that's less than 90%. So if it's less than 90%, don't even look at it. Now we are, thankfully, getting a hit to a terminase small subunit. So terminase small subunit, genome packaging, bacteriophage, DNA binding, viral protein, and this is phage HK97, and it actually is an enterobacteriophage, but it is a phage. The other thing that we should notice is that this is a hit to the protein databank, and we can tell because the protein databank codes have we call them an alphanumeric code. So they have a 4-digit alphanumeric code. So this is 6Z, 6E. They have an _B because some of these proteins have multiple subunits. So sometimes the protein data bank will add this information if it's another subunit. So I can click on this. So if I can click on this, it takes me right to the protein data bank. And there's the 6Z, 6E, and it says crystal structure of HK97 bacteriophage small terminase. So that's pretty compelling evidence that we have a terminase because it's another bacteriophage. But the great part about the PDB is that these are crystal structures. So that means it's not based on any kind of computational result or anything like that. It's an actual crystal structure. And if you want to, I didn't try this. I don't know if this is behind a paywall or not. But that's the DOI. Okay, so you can look at the. Let's see what it is. A lot of times you can find the paper in which they publish these data. There's a lot of other things in the protein data bank I won't get into right now, but you can actually look at the structure of this using different. So we have the biological assembly, and then this is just one of the subunits. So you can play around with that if you like. But if I go back to HHPred, and again look at those top 3 hits. Okay, so this says terminase for this is PFAM. So this is a different database right here. So PFAM is so these are annotations and additionally family information from a range of sources. So this isn't necessarily a crystal structure. And this other hit right here, that is to the UniProt website, and this is a curated website, and there's a gold star here that says that this is reviewed. So there are lots of entries in the UniProt website, and there's a whole team of researchers that go and look through all of the protein sequences that there are in the world and look at them very closely to make sure that the data is as robust as possible. And we can see here that's a hit to a protein. The function's not there, but it's to mycobacteriophage L5. So this is a phage that's in the same family as ours. So we have some really good hits using HHPred. So if I go back to my description, I'm going to say SIF, so again, supporting information for function, HHPred. Top hit is terminase small subunit in phage HK97, and I'm going to provide the protein database code, the 6Z6E, and then I'm going to say 99.68% probability. So I probably won't list the other ones because that's pretty compelling. All right, so if I go back to my slideshow, okay, so where am I? I was a little bit further along. What else? Okay, so we looked at HHPred. Oh, the other thing is, if you could get hits that are 90% that might not be derived from either phage or bacteria. So phage infect bacteria. So sometimes we'll see bacteria genes that are similar. So if there's a hit to Arabidopsis, which is a plant, or human, or yeast, or anything like that, don't report it. So even if you have a 90% hit, if it's not anything that's an organism that is either phage or bacteria, you should not report that. So again, this is just a screenshot of the top hits that I got in HHPred. Okay, so what else can we use? We can use Syntony. So Syntony refers to the order that genes are expressed, and I'm going to click here for the bioinformatics guide that uses Syntony to assign functions. So there's a specific order, especially for the structural genes, which we have at the 5' end. So if we look at this list, we can see terminase is frequently the first gene or close to the first. So we have some compelling evidence from Syntony that this is the terminase. So the other thing that I'm going to do is I'm going to go back to my, I do this for every gene, and I go back to the function list, and I'm gonna use my finder, and I'm going to put terminase in here and see what comes up. And I can see that terminase, comma, small subunit, and you want to do it exactly the way it's done here. So don't use capital letters, make sure the comma is there. But there's another note right here that says if there are not two obvious large and small terminase, then you just have to say terminase. So that means that we need to be aware, are we going to have another gene farther along that's going to be a terminase large subunit? So the other thing I want to do is I want to go to Famirator. So I am signed in, and I want to go to genome maps, and let me also do this for you too. So the other thing we want to do is we want to go back to phagesdb. I'm not sure. Okay, I might just open another tab. Okay, so if I go to phagesdb and go to Landrack. Remember that one of the things we did first is we did a local blast of the whole genome. So if we do this, this tells us the phages that are related to Landrack. And again, the top hit is going to be to itself, but we can see that the alignment isn't perfect, but we can see that we have some closely related phages. So the top hits are to Donovan, T-Chage, Jebex, and A-True Hilltop. So what I like to do is I like to go back to Famirator, and this is a P1 cluster phage. So if I go down to, scroll way down, we have lots and lots of phages these days, to P1. Let's keep going. So I've already done this before, but it'll say that I have 36 selected genomes. So what I do is I just click on that, and it has all 36 of the P1. So I'm going to uncheck that and check the ones I want. So I'm going to do Landrack. So as you see, we're still listed as a draft because we haven't finished our annotation yet. And then we have Donovan and T-Chage, which is here, Jebex, which is here, HU Hilltop. Did I get them all? So I have 1, 2, 3, 4, 5. Okay, so then I'm just going to go up here and then so those are the ones that I want, and I'm going to hit View Map. So if I view map, what happens is it just does it in alphabetical order, but I'm going to want to do this in the order of similarity, and of course I'm going to want Landrack on top. So if I kind of mouse over here, I can bring Landrack up to the top, and so I want Landrack, okay, come on, Landrack up to the top here, and Donovan was next, and then T-Change was after that. So let's put this one back up here. H Hilltop was at the bottom. Okay, so anyway, so you have to do this every time. I don't know if there's a way to say this. Okay, so Landrack, Donovan, T-Change, Jebex, HU Hilltop. Okay, that's good. So I have them all in order. So the purple means that there's a lot of similarity, and if you can, and there usually is on the 5' end, but as you go to the 3' end, if you see other colors or especially white, you'll see that there are differences between our phage and these others. But right now I'm going to look at this is gene number 1. So if I click on this, then I get a number of tabs. So that's gene 1. That's the DNA sequence. That's the protein sequence. That shows us the domains. There's a single domain and it's going to be a terminase. Clusters, this will tell us all of the clusters. So if I click on, say, P1, that will tell me all of the phages in P1 that have the same gene. And there's quite a lot. And it doesn't have function because we haven't assigned it yet. So that's why I like to go down here to Donovan. So if I click on this gene right here, so again, I can see that it's the same fam, 99930, that's the number of the fam, and there's 172 members of that fam. So again, if I click on this one, it'll tell me that's the fam, so I want to make sure I have the same fam. And usually they're color-coded, but you want to make sure that it's the same fam. You also don't want to go by gene 1. You want to make sure that you're looking at the right coordinates, because sometimes the gene numbers aren't the same, I'm not sure. But here we can do the same thing where we have the gene sequence, the protein sequence, the domains, the clusters, and here the function has been assigned. Now that doesn't mean they're going to be in all of them, but if I look at T-Change, it's kind of going off the screen, but you can see T-Change, Jebex, HU Hilltop, every single one of them has the same gene in the same fam, and they've made the functional assignment as terminase small subunit. So I'm going to add that too. So I'm going to say SIF Syntony, terminase is usually the first gene or one of the first. Terminase small subunit is assigned to the same fam and closely related P1 cluster phages, T-Change, Jebex, and Hilltop. So, oh, that was in Donovan too. Let me put that in there. Donovan. Okay, so I'm doing that. I'm hitting post. So if I'm really confident, I'm going to put terminase, comma, small subunit here, and I am confident. I have a lot of evidence to show that I did that right. So let me go back to the PowerPoint. And, okay, so again, Syntony, that's the order. So again, terminase is really the first, so I'm pretty sure I got that right. And this is a screenshot of the closely related phages on Famarator. So you can see, so again, there are no functional assignments on Landrack because we haven't done this, but you can see that we do have, and there he is, there's the terminase large subunit right there. So the functions will be centered over the gene. So we can see that we do have a lot of functional assignments that we can make. Now we shouldn't completely go by Famarator because some of these were done when the functional assignments were maybe using an old list, so we just have to be aware of that. So these are my my notes. I'm actually going to go back to Famarator, sorry, DNA Master, because I just made a few little changes. So one of the things that you can do is, okay, so we're done now. We have a good set of notes for our first gene. because DNA master is finicky, it's a good idea to back up all your notes in a Word document. So okay, I'm going to go back to DNA master. So this is my good set of notes. So again, you're going to want to highlight this, and I'm right clicking where you say copy. So go and open a Word document and copy that in for the first gene, because things happen with DNA master, and you just want to make sure you have a backup. So I would back everything up in Word. So that is, we have called our first gene, and we only have 73 more to go. Okay, so this looks like a long process, and it is, but you will get good at it. I hope this was informative, and I look forward to looking at your gene calls, and have a good day.